From the moment I'm up, it's incredible. Feeling magical. Magical. It's about that time. I say what? Baby, rise and shine. I'm all about that, Yeah, I can feel it, it's about to be a good day. Yeah, I'ma kick it like I never had a bad day. Keep my chin up, that's all. Mama always said like, that's all.
What's up, everybody? I'm Crazy Aunt Lindsay, and you are in the Fab Lab. My Fab Lab professors today are Jael and Nico, and today we're throwing a Juneteenth celebration.
Crazy Aunt Lindsay, what does Juneteenth have to do with science?
I'm so glad you asked. Juneteenth is an American holiday that began on June 19th, 1865. It commemorates the emancipation, or freeing, of enslaved indigenous and imported human beings onto Turtle Island, more closely aligning with the American core value of freedom. Enslavement is horrible. It's a historical practice that human beings have been engaged in for a really long time. It extracts labor and resources from people and land without allowing them to benefit from what they produce. And that's just not the natural order of things. Freedom is, sharing is, reciprocity is. And that's exactly what Juneteenth celebrates. While our projects celebrate the fact that we all deserve to enjoy the fruits of our labor, while also presenting the fabulous opportunity for us to get to know our personal histories with genetics and epigenetics. In this week's episode of the Fab Lab, we're going to extract DNA from a strawberry and meet the double helix, the biological code that has the entire storyline of how I came to be. Then we'll meet an expert and some very high-tech machinery that's going to help us extract that DNA from the strawberry. Then we'll learn some oral history about the strawberry, how it inspired an entire Juneteenth tradition, as well as its connection to the moon. We'll make some sweet treats for our Juneteenth guests: strawberry preserve, Fab Lab community pie, and my very own June Rose, all celebrating community, history, and science. In the Fab Lab Active Hour, we'll shake what our mamas gave us to some classic cookout tunes and co-create a playlist that we'll share together. And then, of course, we'll hit up the Fab Lab library, where we'll read a poem from Maya Angelou, Life Doesn't Frighten Me at All, featuring paintings by one of my favorite artists, Jean-Michel Basquiat. You're invited to the Fab Lab Juneteenth celebration because science is fabulous. The Fab Lab with Crazy Aunt Lindsay, starring real kids from Portland, Oregon. Brought to you by OMSI, this speaker of strawberries, and a team almost entirely made up of neurodivergent creators. Because science is fabulous and also for everyone. Science says that we learn best not just when we're having fun, but when we're relaxed. We're gonna take the Fab Lab heartbeat together to center our minds and our bodies to get our brains ready to learn about genetics. Close your eyes, put your hands over your heart, take a deep breath in, and a deep breath out with a sound. Nice. Now for a count of 4, we're going to inhale. 1, 2, 3, 4, hold. 2, 3, 4, exhale. 2, 3, 4.
I'm ready to learn about genetics.
Great. Let's learn about genetics. Genetics is the study of our genes. No, not those kind of genes, these kinds. The ones we inherit from the people who came before us. If you ever wondered why you keep hearing people say, "Oh, you look just like your mom," or "You got that nose from your grandpa," that's because they gave you their genes, the natural biological instructions or code that determines all sorts of traits like your eye color, hair texture, blood type, where your nose goes, and even your nutritional needs. Take my family, for instance. This is my mom, and this is my dad, and this is my dad's mom. There's no mistaking who I come from, especially not when I get to laughing. All that information starts with and lives in our DNA. Deoxyribonucleic acid is what DNA stands for, the molecules that form the double helix, a swirling ladder of rungs and packages that hold all the information that form you, me, and every living thing from strawberries to insects to human beings. DNA acts like an instruction manual that tells organisms what and how to become. It's a magical, beautiful line of code that unpacks itself into all we are. Rosalind Franklin is the woman who helped first identify the double helix, the structure of DNA. Scientists already theorized or thought that DNA had a helical structure, working that corkscrew vibe. But most of them couldn't agree on whether it was 1, 2, or 3 strands. But Auntie Rosalind's photo 51, a very special X-ray photograph that required over 100 hours of exposure to make, provided everything she needed to calculate and prove there were 2. Some years after she died, big portions of her work contributed to the earning of the Nobel Prize for Watson and Crick in 1962, as well as another Nobel awarded to one of her other colleagues in 1980. But I'll save that one for the blog post on thefablab.com. One thing is crystal clear. Rosalind revolutionized medicine, biology, and agriculture, and is known as the unsung mother of the double helix in genetics. Which leads us to another unsung scientific hero, Dr. E.E. Just, known as the forgotten father of epigenetics, one of the most revolutionary developments in current biological sciences. Epigenetics is the study of how our behaviors and environment can cause changes to our DNA that affect the way our genes work, express themselves, and are passed on to our children. And also how our parents, grandparents, and ancestors' environments, behaviors, stressors, diet, trauma, and beliefs may have left reversible impacts on our DNA. The fact that these impacts are scientifically proven to be reversible means that with a little awareness, understanding, and effort, we can heal them. Way back in the early 1900s, Dr. Ernest Everett Just was a cellular biologist and professor of biology and zoology at Howard University in Washington, D.C., where he specialized in cytology, the study of the cell. To this day, his name is almost never mentioned in textbooks or lecture classes. But the study of gene expression was sparked by Dr. Just over 100 years ago when he developed his theory on what he calls genetic restriction. And my goodness, was he handsome. Anyway, read more about Uncle E and Auntie Rosalind, genetics, and today's projects at thefablab.com. Now, we're going to extract DNA from a strawberry. For this project, all you're going to need is a few things that you definitely have laying around the house, with the exception of maybe the thing we're going to extract from, which is the strawberries. So go out and get those. For this project, you're going to need strawberries, rubbing alcohol, salt, dish detergent, distilled water, and quite a few cups. Oh, and don't forget about your coffee filter. So all you're going to do is take your strawberry bag. Here's your strawberry bag. And now comes the really fun part.
Smashing them?
Let's smash them.
Yeah.
Be careful.
This is fun.
Make sure you're breaking up all your strawberries.
Ow.
Boop, boop, boop. So for the DNA extracting solution, all you need is 2 tablespoons of dish soap, a teaspoon of salt, a half a cup of distilled water. This is your DNA extraction solution. So now what I'm gonna do is I'm gonna close this up very carefully and do my best to limit the amount of air and bubbles that I create. Nice. And then very tenderly mix it up like this. Science is messy. We're going to become human centrifuges. A centrifuge is a very, very high-tech machine that scientists use to further isolate genetic material. They spin so super fast. But before we had the technology to make that possible, we did something really simple. This, friends, is about to be your centrifuge. Nice. Perfect. Nice job. You're doing so good, yes. We're gonna do something called precipitate alcohol into our solution. Can you see what's happening?
It's getting silvery on the, like a layer on top.
It's getting a what on top?
Silvery layer.
Oh, what do you think that silver layer is?
Alcohol.
No. That, friend, is DNA.
What?
Ah!
Oh, good lord.
Yes!
That's sweet.
That, friends, is isolated double helixes all folded in on one another. I can't wait to see what it looks like up close.
Yeah.
Recognize this? Yep, it is our strawberry goop from our DNA extraction project. I'm here with my friend Shannon from OMSI, and she's going to introduce us to a very high-tech piece of equipment where we're going to get up close and personal with our strawberry DNA.
All right, so right here we have our sample of strawberry DNA.
Sample?
Kind of try to look at it and see what you think you're looking at.
Super scientific word, observing the sample.
Yes.
I am observing a very delightful strawberry scent
Okay.
with a hint of alcohol.
Yes.
I'm also seeing a lot of seeds. I'm seeing liquid. I'm seeing some white stuff on
Right.
top that I am gonna call membrane.
Yes. So when I made this, I had to use strawberries and pineapple juice in a bag, and I had to make sure I mushed them up really nicely. And then I added alcohol on the top. So if you try this at home, make sure that the alcohol is only on the top, because these white things that are forming right here, that is our DNA that we're looking for.
The bubbles and the goopy stuff
Yes.
that's sort of like white.
Right.
Okay, awesome. I love this. What does
you.
it look like in this contraption?
Isn't this a contraption? Well, first we're gonna look at it in our eyes. Do you see anything in there that kind of looks two-strand-ish?
Definitely I'm not seeing two strands.
No. How about this tool here? This is a magnifying glass. Give that a try.
I know this. I'm seeing more bubbles.
Okay.
That is it.
That's it. Still not strong enough. So this here is a microscope.
It is very intimidating.
It is.
What kind of microscope is this?
So this is actually a simple microscope, but there is not many things that are simple about this. This microscope can do many amazing things.
Very cool.
So we are going to try to see the strawberry DNA with this microscope.
What does this do that
course.
makes it possible for you to see this up close?
So these pieces here will help us to magnify. Magnify means to make bigger on this microscope. So first we're gonna put our slide on this stage, and this is called a stage. Why do you think it's called a stage?
Because I'm seeing lights, I'm seeing cameras, and I
Okay.
know there's some action about to go down.
That's right, this is the spotlight where the action happens. So we are going to slide our slide on the microscope.
Nice.
And for the parts, these knobs right here help me to move my slide back and forth. And this helps me to move my object to the left and to the right so that I can try to see it through my eyepieces. I still can't see anything, so I'm gonna remove my glasses because the microscope helps my eyes to see the magnification. These knobs on this side help me to move my object a bit closer. Would you like to look?
I would love to look. Let's see. Ooh, I see a lot of white.
Okay.
I can see, oh my gosh, I can see even more bubbles up close. And I'm seeing some bigger bubbles.
Yes.
I'm seeing more debris that I think is like some of that like packaging just sort of like coming off to start letting things come out.
But still no DNA.
Still
huh?
no DNA. Is there anything else that we can look at that we might be able to see a little more?
That's a great suggestion. Sometimes in science, the first thing that we try just doesn't work out. So we can have a backup plan. So here we are going to use a leaf. So this here is our leaf. If you hold it up, you can actually kind of see it through the light. So this will work better on our microscope. Rearrange.
This is so
we can
cool.
look through.
I can see some like big veins in the
Yes.
leaf. I wonder what they look like up close.
Yes.
I am using, oh my gosh.
Discovery.
I mean, literally it looks like
Yes.
like a green desert.
Yes.
Like, and I'm seeing veins all over the place and I'm seeing these almost like buttons of beautiful purple.
So what we see there are cells. So just a reminder that this is a living plant. So thank you, science, for contributing to our experiment today. Plants are alive. So what you can see here are the plant cells.
And inside of the cells are where the DNA lives.
Yes, that is where the DNA
Very
is.
cool. And the muddling or the crushing up helps to get it out.
Yes.
Very cool. Will you talk to me a little bit about why microscopes are important? Why is it so important that we look up close
Yes.
at things like leaves and DNA?
Yes, so when we're making new discoveries, it's important that we can really take a good look at what we're trying to see. So a chemist is not the only person that uses a microscope. For instance, a forensic scientist might use a microscope to look at fingerprints, or more importantly, a genealogist will look at this. So if you ever are in DC, you can visit the National Museum of African American History and Culture. They have a family lab there with genealogy that you can look up your family history.
Wow.
Yes.
Thank you so much, Anna. This was so cool.
Woo!
There's the science. Now let's make it fabulous. Now it's time to make Fab Lab strawberry preserve. Nice, half the sugar in, nice. Pour your water on top. You're going to add your preservative. Lemon hosts citric acid. It's what gives lemon its sour taste, but it's also a natural preservative that's been used for thousands of years. Have an adult cook this on a stovetop for about 30 minutes. And then because this is YouTube magic, we have our strawberry preserve. Next, we're gonna make the Fab Lab June Rose. All you're going to do is add some muddled or preserved strawberries to ginger ale or sparkling water. Dress it up with some mint and a splash of rose water if you happen to have some laying around, and voila! Next is the Fab Lab Community Pie. This one uses fruit to help celebrate the diversity in all of us. The colors for the Juneteenth holiday are often associated with red, white, and blue, which represents that the enslaved indigenous and imported people who were forced to work here are American. But the colors that I grew up associating with Juneteenth are green, red, yellow, and black. Black for the color of the skin of the people who are mostly enslaved. Red and green to feature the prosperity. Yellow is the gold that lives inside of all of us. We're going to make a community pie that celebrates all the different meanings that you might come up with. We're using pre-made Fab Lab sweet cream and scone dough shaped like a pizza as the foundation for our community pie. Next, we're going to do, of course, our strawberries. This is so cute. Okay, the next thing we're gonna add is our yellow. Nice. Perfect, this is so cute.
Today. Oh, let me put one more,
One more?
actually.
Yes, right there. Just a last touch, a little lemon zest. Ooh, that
Oh, that smells good.
It smells so good. There it is, our Fab Lab community pie. We're gonna go set this on our Fab Lab celebration table and see you there. For this week's Active Hour, we're working off our celebratory sugar rush with dance. I grew up listening to some iconic jams that were always featured at our family gatherings and Saturday morning chores. My Fab Lab professors are going to teach me some new moves as we listen to the playlist that I created for this very occasion. Check it out and add your favorite family jams to the Fab Lab Juneteenth celebration playlist on Spotify. And make sure you tag me on Instagram and TikTok with the dances you create. Thank you. Life Doesn't Frighten Me by Maya Angelou is this week's Kid Book Club pick. Maya Angelou is the Grammy-winning civil rights activist, poet, and author who received the Presidential Medal of Freedom in 2010 from President Obama. She's famously known for her honest and uplifting words and spirit, which is part of why I chose to read this book to you guys for this week's Juneteenth celebration. Life Doesn't Scare Me. Another reason that I chose this book is because it features paintings by one of my favorite artists, Jean-Michel Basquiat, the Haitian-American abstract expressionist and graffiti artist who roamed the streets of New York in the 1980s, a pretty scary time in New York, and was really good friends with Andy Warhol, another famous artist. Let's read and discover why some pretty scary things don't frighten Aunt Maya and Uncle Jean-Michel. Shadows on the wall, noises down the hall. Life doesn't frighten me at all. Head over to Patreon and read this whole story with me. Life doesn't frighten me at all. Not at all, not at all. The end. That's Maya Angelou. How'd you guys like that?
It was pretty good. I loved it.
It's pretty good? Dope. You can learn more about Maya and her work on the Fab Lab library shelf at thefablab.com. I mentioned earlier that strawberry soda is a staple tradition for the Juneteenth holiday across the country and is the inspiration behind our fabulous June Rose. I found it curious that strawberry soda was a thing for Juneteenth because strawberry soda hadn't even been invented until 1907, becoming commercially available in the 1960s, almost 100 years after the freeing of enslaved Americans. So I did a little research, and I found out some really interesting history. Did you know that every full moon has a name? January's is called the wolf moon because that's when they started to howl outside of villages because they were hungry from the slim pickings of winter. March is called the worm moon because that's when the ground started to warm and they started wiggling, signaling the start of spring. And May is called the flower moon because that's when we can observe they start to bloom. The names of every full moon that we know of from the Farmer's Almanac, a 300-year-old publication that helped farmers with very important jobs like predicting the weather before TV and weather apps were invented, and was actually founded and published in my hometown of Marstown, New Jersey, thank you, were all names given to the moons by the Algonquin tribe, one of the indigenous peoples on Turtle Island, what we now know as the United States of America. They gave them their names because of the patterns that they observed as the seasons changed and what was happening in nature at that particular time. Can you guess what the full moon in June is? Very close.
Strawberry moon.
The strawberry moon, yes. It's called that because June is the peak harvest season for strawberries, which aren't even berries at all. They're actually multiple fruits clustered up together into one, coming from the rose plant, which is why I call this the June Rose. The full strawberry moon of June 1865 would have happened on the 9th, 10 days before Juneteenth, meaning that at the time of our ancestors' good news of freedom, they would have been right in the middle of the strawberry season with an abundance of strawberries around them, making it very easy to make that their celebratory treat. My hypothesis is that this is why strawberry soda is our celebratory companion for every Juneteenth holiday. What an awesome celebration. You already know that I have a ton of people and things to thank. First, I'd like to thank my parents, my grandparents, and ancestors for all they endured so I could be here. And thank you to the strawberry for helping us not just celebrate, but see the DNA that holds our living story. Also, to whoever invented the centrifuge for saving so many arms and probably a few noggins. Thank you to Rosalind Franklin for her work identifying the double helix and dealing with her super rude colleagues. And E.E. Just for thinking the revolutionary thoughts that led to the study of epigenetics. I'd like to thank Maya Angelou for her words and spirit of freedom. Jean-Michel Basquiat for those amazing paintings. I'd like to thank my professors, Jael, Nico, Arthur, Esme, Mila, and Miles for being such great learners. And I'll thank myself for doing my best to fearlessly persevere through the challenges that can come with learning about history and science and sharing it with all of you. And of course, thank all of you for being with me today, because science is fabulous, and so are you. Thank you to OMSI, the Marie Landrum Foundation, and the entire Fab Lab community of patrons on Patreon and iFund Women for making this entire season of STEM programming possible.